Rapamycin and other mTOR inhibitors represent a breakthrough in the science of aging, not because they directly lengthen telomeres, but because they slow the processes that cause telomeres to erode and cells to senesce in the first place. The mechanistic target of rapamycin mTOR pathway is a central regulator of cell growth, nutrient sensing, and protein synthesis. Under conditions of chronic stimulation, such as from excess calories, persistent inflammation, or metabolic stress, mTOR remains highly active, which accelerates cellular aging, impairs autophagy, and increases the risk of age-related disease. Inhibiting this pathway, even periodically, appears to restore a more youthful balance in cellular signaling, allowing for improved repair, stress resistance, and reduced senescence. Rapamycin, originally discovered as an antifungal compound on Easter Island soil, has been shown to extend lifespan in multiple species, from yeast and worms to mice, and potentially even dogs. The mechanism is multifactorial, but central to its anti-aging effects is the promotion of autophagy, the cellular housekeeping process that clears damaged proteins, dysfunctional organelles, and other debris. This cleanup process not only improves cellular efficiency and resilience, but also reduces the burden on telomeres, which are vulnerable to oxidative stress and metabolic dysfunction. While rapamycin doesn't directly activate telomerase or elongate telomeres, its ability to suppress the chronic activation of the mTOR pathway significantly reduces the rate at which cells enter senescence. Telomere erosion is one of the key triggers for a cell to become senescent, essentially alive but non-dividing and functionally impaired. Senescent cells release inflammatory signals, known as the senescence-associated secretory phenotype, or SASP, which further accelerate aging in neighboring cells. By reducing the accumulation of these dysfunctional cells, mTOR inhibitors like rapamycin help maintain a healthier tissue environment and may indirectly protect telomere integrity over time. There's also growing evidence that mTOR inhibition improves stem cell function. In aged tissues, stem cells often become either depleted or inactive, in part due to telomere shortening and the metabolic burden of persistent mTOR signaling. Rapamycin appears to rejuvenate these cells, supporting better regeneration in muscle, immune, and even neural tissues. This regenerative effect is vital in maintaining the body's ability to heal and adapt with age, and it likely reduces the demand on existing somatic cells to replicate, which would otherwise lead to more telomere loss. In addition, rapamycin has systemic effects that support telomere health indirectly. It improves insulin sensitivity, lowers chronic inflammation, and reduces oxidative stress, all major contributors to telomere damage. In animal studies, rapamycin-treated models show reduced incidence of cancer, cardiovascular disease, and neurodegeneration, conditions that have also been linked to accelerated telomere shortening. These broad-spectrum protective effects suggest that, even without altering telomerase directly, rapamycin helps sustain a biological environment in which telomeres erode more slowly. Human trials are now cautiously exploring rapamycin analogs, rapalogs, for aging-related applications. Low-dose and intermittent dosing strategies are being tested to retain the benefits without causing immune suppression or metabolic dysregulation, which can occur at higher or continuous doses. Initial results are promising. Older adults taking low-dose rapalogs have shown improved vaccine responses, reduced inflammation, and even signs of enhanced physical function, all pointing to a slowed aging process at the cellular level. What's particularly exciting is the compatibility of mTOR inhibitors with other longevity interventions. For example, when combined with intermittent fasting, which also downregulates mTOR, there appears to be a synergistic effect. Likewise, mTOR inhibition may complement telomerase-based therapies by reducing the cellular stress that drives telomere erosion in the first place. This points to a future where anti-aging regimens are personalized, combining interventions that slow wear and tear with those that restore cellular capacity. In summary, while rapamycin does not directly extend telomeres, its powerful modulation of mTOR signaling allows cells to age more slowly, repair themselves more effectively, and avoid premature senescence. This protective environment slows telomere loss indirectly and maintains genomic stability, making rapamycin one of the most promising pharmacological tools for extending health span. If future human studies continue to support its safety and efficacy, mTOR inhibition could play a central role in future anti-aging protocols, not by forcing cells to divide indefinitely, but by helping them thrive longer and more functionally before reaching their replicative limits.